Good evening, everyone. Welcome to Monday. Finally, we are meeting after 2 days off, and I hope you also are like triggering to meet to learn more, to have more learning point of view. It's already 1908, so we start by 1910 to finish off with our protein purification. So today, earlier days we were doing with a lot of chromatography, HPLC, gel electrophoresis, SDS page, how to prepare samples for 2D electrophoresis. So today we will understand about how 2D electrophoresis is done in reality. So that will be today's task for today. And please follow me on LinkedIn. So I will start uploading new things on it. So this is my LinkedIn network. It's very for those who doesn't know about it, it's a very good platform to make networking for your future jobs or any other certificates or any things that you're looking professionally to build up your good connections. It's one of the best I would say platform. So go for it. This is my LinkedIn profile. I will share in the Telegram group. Also, for those students who doesn't have subscribed to my channel, please subscribe. So I'm also on Instagram. Sometimes I used to upload things of my doing exercise and gym, but now I have stopped it. This link. Please do subscribe also. I think we can start now. It's already 9 or 10. We should not waste much of our time. So how many students do we? It's getting very less. I don't know why. Every time I come here, it's reduces. It's 30. Okay, we can start. We can start. Okay, somebody send me some requests on it. So after your samples were prepared in the 2D dimensional gel, last time we did. So what will happen? Your protein part, who has isoelectric point to be 0 somewhere, pH is 4.0, pH is 10. Your protein mixture will flow from lower to higher. So pH 4.0 to pH 10.0. And so in the first dimension, they will separate it because of charge. And after the pH thing is over, then we separate them by size, according to the size. First by charge, then by size. So we will put them on another gel, and you will have various spots of different proteins according to your SDS electrophoresis. So here you can see an example showing from the JVC paper that isoelectric focusing with the SDS page producing lots of spots and producing many multifold proteome of one organisms within this species. So we have so many proteins in our body, and we can actually recognize each of them within this proteome with the help of 2D electrophoresis. So let's see how it is done practically.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the Ipg strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality two-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, chaps, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has two sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip. On the negative side, you'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before the step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip